Hi guys, Dr. Orlando here with a vidal about light reactions of photosynthesis. This is just like we talked about in class and exactly what we drew on the board. So you should be able to follow along in your notes and make sure that you've understood this fully. Okay, so where are we in this cell? Move this over a little bit. Where are we? We are, well, where are we in the plant? We're in a leaf. We are in the mesophyll, which is that middle layer, the business part as I called it, of the leaf. We are in a cell. Move that over. We're in a cell. We are in a chloroplast. We are in the thylakoid. Remember, those are the things that look like coins stacked inside of the chloroplast, and it's really the business part of the chloroplast. And specifically, we are looking at the stroma, which is outside of the thylakoid membrane, and then the thylakoid space. So we have zoomed in so much, so this is all we can see in the picture that I've drawn. So let's go ahead and label those areas. We're going to label stroma, thylakoid membrane, and thylakoid space. Okay, so out here is stroma, the space around the thylakoid. This is the, move this over, thylakoid membrane. Okay, and then in here is the erase this, time for a new stylus. Thylakoid space. Okay. All right, so we have our where we're at in the cell and in the plant figured out, right? So the sunlight shines in through the leaf, and it's going to affect what's going on here in the thylakoid membrane. First thing that's going to happen is water is going to be split. Okay, right here. Check. Okay, so we're going to, actually let's do numbers like we did in class. So the first thing that's going to happen is water is going to be split, and it's split into oxygen and hydrogen, and an electron is essentially lost from the splitting of water. That electron goes into this first area here. It's all very colorful, and we call this P PS2, and it's photosystem 2. I know 2 comes before 1. It's just the way they figured them out, and then they figured out their order. So photosystem 2 is the first stop for this electron. And that photosystem 2 is made up of pigments. The light green ones in my picture would be chlorophyll, and then we'd have our xanthophylls and carotenoids and different color pigments in there that are absorbing light from the visible spectrum of light. We absorb red, blue, purple. Remember, the reason that we see it as green is because green comes in and bounces back, and so our eyes can see it. That's supposed to be an eyeball. Get lashes. Okay, so we see that one, but it's going to absorb the rest of the spectrum, and when it does that, it's going to energize this electron. These orange little dashes are showing that the electron is energized. Okay, so now that the electron has high energy, it has absorbed the photons from the sun. It's going to jump from photosystem 2 to the electron transport chain. And remember, I said these guys are an electron transport chain 2. Okay. These guys are proteins. They are membrane proteins. I draw them like these little factory workers with their hard hats because I want you to remember that the proteins are the hard workers here in the membranes. And in this case, their job is to take that electron. I'm going to draw him yellow so that he shows up well. Take that electron and pass it along. And as it's being passed along, the energy from that electron, these really high energy to begin with, the energy from that electron is going to be used to do something. And in this case, see, I'm going to draw less little yellow dashes around him so you can tell he's losing energy. The energy from passing him along is going to be used to change the shape of these proteins, of my worker guys here, and they're going to become a pump for hydrogen ions. Whoop. So there's a bunch of hydrogen ions out here. So these hydrogen ions are going to be pumped from the stroma into the thylakoid space. Okay? And we're going to create a hydrogen ion concentration gradient. Concentration. My goodness. I'm having a hard time getting my colors to switch here. So we're going to create a hydrogen ion concentration gradient inside. Why do we want to do that? Well, a lot of times concentration gradients are used to drive pumps, okay, or to drive other processes. So think of this hydrogen ion concentration like a potential current. So maybe think of it like water that could go over a water wheel or a turbine to make it spin. So as soon as we open the floodgates, the water's going to run over the wheel and power something. Well, that's what's going to happen here. As soon as we open the floodgates, the hydrogen ions are going to go out. What are they going to go out? They're going to go out of this guy here. This is called ATP synthase. Okay, remember ACE at the end of a word means that it's an enzyme. This is going to synthesize ATP. That's its job. This is an enzyme. It is a protein. It's embedded in the thylakoid membrane, and it will act almost like a turbine. It actually spins. So as these hydrogen ions flow through ATP synthase, and they'll happily go down their concentration gradient, right? Because we created this gradient. They will happily go down that concentration gradient. It actually causes the ATP synthase to move. And when it does that action, it will take ADP plus an inorganic phosphate, and it will turn it into ATP. Not turn it into, it'll take that phosphate and add it to ADP and make ATP, so adenosine triphosphate. So now it's energized. We have our energy carrier made. Remember these terms, energy carrier. Okay. We have the energy carrier made, and lots of it, lots and lots of ATP. So this first step here, we started with the photosystem 2, and that electron was used to pump the hydrogens through the electron transport chain. The hydrogens were used to drive ATP synthase, an enzyme that converts ADP and phosphate to ATP.
So we made lots of ATP. Now we take a step back over here and we say, well, what happens to that electron? Well, now he's a low energy electron. Okay, he doesn't have much energy at all. So he has to get recharged. How are we going to do that? Well, let's put him into another photosystem. That sounds like a good idea. So we put him in there. The sun shines on this photosystem. This is PS1, photosystem 1. Sun shines on photosystem 1. Remember, again, these photosystems are where we find the pigments like chlorophyll and xanthophyll and carotenes, or carotenoids. And so we've got lots of colors in there that could absorb the different wavelengths of light from the sun. Sunlight shining there. And then the electron is energized. And again, he gets so energized that he jumps out of photosystem 1 and he jumps over to the next electron transport chain and he goes to ETC. Oop. Get rid of that there. He goes to ETC1. That's Roman numeral number 1. So ETC1, which again, the electron transport chains are a series of proteins embedded in the thylakoid membrane. They work together to do a job, just like factory workers work together to do a job. So that's why I draw them looking like factory workers. Okay, so the electron is passed along through this set of proteins, and it is on the last step. On the last step, that electron will be loaded, as I said, into the backpacks of the electron carriers. What does that mean? Well, remember, we have electron carriers that can move electrons around for use in pathways that require energy. Okay, so we have NADP+. And remember, I drew this as the guy, I'm going to zoom in here so I can draw a little better. The guy with the backpack, and he's sad because his backpack is empty, right? So NAD+. So what happens is that electron is loaded into the backpack, and now he's NADP. He loses the positive charge because he's going to get an electron. He's also going to get a hydrogen. Electrons and hydrogens are friends. It makes sense. The hydrogens have a positive charge and the electrons have a negative charge. And so here he is, our electron carrier. He's happy because he has an electron in there and he's got a hydrogen. Okay. So we made from this second step, or the second part of our photosystems here, we made an electron carrier. Lots of them. Okay. All right. Okay, so to sum up, from photosystem 2, we have produced lots of ATP. And from photosystem 1, we have produced lots of NADPH. Okay. ATP is our energy carrier. And the energy is carried in the bonds, the covalent bonds of the phosphates. The NADPH is our electron carrier. And remember, I drew these guys like a guy with a backpack, and he's going to carry that electron along with a hydrogen ion to another process within the cell. And that's what we will be talking about next. All right, if you have any questions, please let me know. Have a great day.